last presentation of this session. The last speaker is Professor Olson from University of Texas Southwestern Medical Center. The title of his presentation is The Molecules and the Mechanisms of Heart Development.
Okay, I'd like to thank the organizers for inviting me to this meeting and thank all of you who've stayed to the end. What I'm going to do today is to talk to you about one short story on some recent work from our group which has focused on mechanisms and new approaches for heart regeneration. So our group over the past many years has used muscle cells, skeletal muscle, cardiac, and smooth muscle cells as model systems for deciphering fundamental mechanisms of development and gene regulation, and along the way we've uncovered many transcriptional regulatory circuits and important roles for microRNAs in the control of development and disease of these various muscle tissues. So today I'm going to talk to you, just as I said, about one short story which focuses on the heart and strategies for regenerating the heart in response to injury. So the heart is, as I think many of you appreciate, the most effective, really astonishing pump ever invented. The heart is the first organ to form and function in the embryo, and all subsequent events of life of the embryo and the adult require its second-to-second function. Just to put the power of this organ in perspective, if you keep in mind that the adult human heart weighs only 11 ounces and it pumps 8 tons of blood every single day, the average human heart will beat 3 billion times in a lifetime without ever skipping a beat. In fact, it only skips one beat. That's the last beat. And the heart, though, despite this power, has a fundamental flaw, and that is that the heart is extremely complex and fragile and is susceptible to malfunction. And as a consequence, congenital heart disease, seen in 1% of newborns, is the most common human birth defect. And I think all of us are aware that adult heart disease is the number one cause of morbidity and mortality, exceeding all cancers combined. So there is an enormous need to not only understand the fundamental biology of how the heart forms and functions, but also to design new strategies for heart repair. This slide summarizes the fundamental problem, and this is really the number one biomedical problem in the world today, and that is that the heart cannot regenerate or repair itself following injury, and the default response of the adult heart to injury is fibrosis. So this is a histological section through a mouse heart, but a human heart would look much the same. This is the chamber of the left ventricle, which pumps blood to the body. This is the right ventricle. In response to a coronary artery occlusion during a myocardial infarction, or in response to many other cardiac disorders, there's a massive and almost instantaneous loss of myocytes from the left ventricle of the heart, which is the primary pumping chamber, and these are replaced by a fibrotic scar. You can see here. The scar serves as a barrier to regeneration, and it lacks any contractility, and as a result, the heart loses effectiveness, it dilates, and ultimately progresses to heart failure and death. There have been thousands of patients treated over the past decade in the United States and Europe with stem cell therapy to try to enhance function to the failing human heart. And I think it's fair to say that all of those studies have failed, and so far there's no evidence for injection of stem cells into the heart that can give rise to new cardiac muscle cells. And so I think it's time to really take a step back from this problem and think about ways to leverage our understanding of developmental mechanisms as a strategy for generating new heart muscle. And so what I'd like to do today is to talk to you about some of the really new approaches and some of the ideas that we're trying to apply to this problem. Now, there's nothing inherent in the heart per se that is incompatible with regeneration. And in fact, some organisms have solved this problem. For example, zebrafish and many types of amphibians, newts, salamanders can regenerate large portions of their heart following injury, as they can also regenerate organs. But clearly the adult mammalian heart whether it be a mouse or a human, lacks any appreciable regenerative potential. So we decided to ask a question that surprisingly had not been asked previously, and that was, might there be a time in development of mammals when there could be the potential to regenerate portions of the heart? And if we could identify any regenerative window, might we be able to amplify those mechanisms in the setting of adult regeneration? And so we initiated these studies in mice, and we began during the neonatal period and developed methods where we could injure the neonatal mouse heart and monitor its response to injury. And when we did these experiments, we were stunned to find that the neonatal mouse heart can fully regenerate following injury. And this is a typical experiment. What we would do is perform an apical resection where one can amputate 20% or so of the mass of the heart at the apex. And if one then allows these animals will survive this surgery and they can be refostered with their mother. And if you monitor histologically what takes place in these hearts following injury, there's an immediate inflammatory response which has to seal the chamber of the left ventricle, or the animals will exsanguinate. Then there's inflammatory response, there's proliferation of muscle cells across the organ, and ultimately there's complete disappearance of the wound and restoration of function. These animals, following massive injury to their hearts, can fully regenerate cardiac function and structure, and they then go on and live. They can live the same lifespan as a normal mouse, and they're completely indistinguishable. So the amount of tissue mass that was removed in these initial amputation experiments, roughly 20% of the mass, is the same amount of tissue mass that's been used in studies from zebrafish and salamanders, which documented the regenerative potential of those organisms. Now, surgical amputation is obviously not the primary stimulus for heart injury in humans, and so we wanted to ask whether inducing a massive myocardial infarction in a neonatal mouse could also be followed by a regenerative response. And so we developed methods for tying off the coronary arteries to the heart and inducing a myocardial infarction. And the way this was done in these experiments was we would induce an infarct by tying off the artery, and we began on day 1 after birth, and we look at the animals 21 days later on day 22. And these are cut in cross-section in contrast to the previous slides, which were longitudinal. What you can see here is that if you induce a myocardial infarction at day 1 and you look 21 days later, there's no evidence for a myocardial infarction in these hearts, indicating that they have fully regenerated all of the ventricular myocytes following injury. If we delay the injury to day 7 and induce an MI and then look 21 days later, we can see quite a bit of evidence of regeneration, but there's also a prominent scar forming that you can see here. And if we allow these animals to go to 14 days or to adulthood, they exhibit the adult response to cardiac injury. That is, that there's no regeneration, and instead there's massive formation of scar, a blockade to contractility, and dilation of the left ventricular chamber. So from these experiments and others, we've concluded that the ability of the mammalian heart to regenerate is impeded by the process of fibrosis, and the fibrotic response becomes more robust with time as the regenerative response declines, and ultimately by the adult stage, fibrosis occurs maximally and regeneration is completely prevented. Now, we've gone on and have characterized the cellular processes associated with the neonatal regenerative response, and they are much the same as those seen in more primitive organisms like zebrafish and amphibians. So the initial response is an inflammatory infiltration in the region of the lesion. There's activation of a layer of cells surrounding the heart known as the epicardium. I won't talk about this today, but this is an injury-sensitive stem cell niche that's activated in response to injury and provides signals for regeneration. There's new blood vessel formation, and there's a fascinating response that we're still trying to understand, and that is that cardiac muscle cells across the organ, from top to bottom, are induced to proliferate when there's an injury at the apex, and we're currently trying to understand if that's a mechanical or a biochemical signal that induces these responses. The culmination of these events is complete regeneration of the heart. and each of these events is blocked in the adult heart. So just to look a bit deeper at some of these steps, here we've monitored the process of new blood vessel formation by casting. All right, let's hope for the best here. All right, so here we've casted the complete vasculature in the heart following injury. If we induce a coronary artery ligation right here, this entire region of the heart would die. This is the zone of regeneration. And what you can see is during the neonatal period, there's massive formation of new blood vessels, both from left to right, left to left, and right to left. Now, we've wanted to understand what is the cellular and molecular basis of new blood vessel formation, and we've investigated the inflammatory response. We noticed early on that macrophages infiltrated into the heart following injury, and so we've eliminated the macrophage population by injecting animals with liposomes impregnated with clodronate, which can be phagocytosed by the macrophages. So here again is a neonatal heart. We've induced a myocardial infarction. Look, 22 days later, it's regenerated. There's just a trace of the infarct. If we eliminate the macrophage population, regeneration is completely abolished, and instead we have scar formation. So macrophage infiltration during the inflammatory response is clearly critical for neonatal regeneration. We can look more closely at this, and this may be a bit hard to see, but here's a regenerating heart. In the infarct zone, you can see all these new blood vessels here stained for PCAM, seen here also in red. And if we eliminate the macrophage population, there is no new blood vessel formation, and instead there's simply a fibrotic scar devoid of new blood vessels. So from these and other experiments, we now believe that the injury response to the neonatal heart leads to an innate immune response in which macrophages and other types of inflammatory cells infiltrate the injury, and they release a number of cytokines, which I won't have time to talk about today, that we believe stimulate neoangiogenesis and probably survival of muscle cells, contributing to regeneration. In the adult heart, this step is blocked. There's a different spectrum of macrophages that infiltrate the heart and a different constellation of cytokines released, which we think drives fibrosis. So these, again, are the steps involved in neonatal heart regeneration. One of the other important steps that I described is that cardiac muscle cells are induced to proliferate across the organ, and very little is known about mechanisms that can drive proliferation of cardiac muscle cells. Norbert Perimon described an ancient signaling system that controls cell proliferation in organisms ranging from fruit flies to mammals. This is known as the Hippo pathway, which is comprised of a series of kinases and adapter proteins that transmit growth signals from the cytoplasm and the exterior of the cell to the nucleus. The terminal transcription factor in this pathway is a transcription factor called YAP in mammals and Yorkie in fruit flies. And so we tested whether this pathway might be involved in the control of cardiomyocyte proliferation. If we elevate the expression just of this transcription factor at the end of the Hippo pathway in the embryonic heart, shown here in a transgenic mouse, you can see that simply expressing activated YAP is sufficient to drive the production of excessive cardiac muscle cells here compared to a control. So we then used a genetic strategy where we could selectively activate. This is going to get old real fast here. All right. Let's try again. We selectively activated YAP in the adult heart. And induced a myocardial infarction. In a normal heart, if you induce an MI, you have fibrosis and dilation with, this is sequential sections across the heart. In animals that have expression of activated YAP in their heart, they're resistant to this injury. They have excessive proliferation and survival, and you can see here they retain heart muscle following injury. If we measure the size of the fibrotic scar following injury in wild type compared to transgenic mice, you can see that the fibrotic scar is diminished when we simply activate this terminal step in the Hippo pathway. And this is a bit complicated, but if you measure cardiac contractility In normal mice, when you induce a myocardial infarction, contractility drops precipitously, whereas in animals with activated YAP at the end of the hippo pathway, they maintain cardiac contractility due to proliferation. So what I've told you so far is that that there are several events that enable the neonatal heart to regenerate, and those steps are blocked or diminished in the adult heart. And so we're currently trying to manipulate those pathways using microRNAs, regenerative genes, or regenerative molecules. Now, in the last few minutes of the talk, I'd like to tell you about a complementary strategy, and that is to reprogram the fate of fibroblasts, which would otherwise form a scar, into new cardiac muscle cells. Now, while we're most familiar with cardiac muscle cells as the primary cell type of the heart, it turns out that roughly half of the cells of the heart are not cardiac muscle cells, but are instead fibroblasts, which serve as interstitial cells linking cardiac muscle cells and other cell types, and also serving as signaling centers in the heart. Now, in response to injury, when myocytes are lost, as I've described, the fibroblast population becomes activated and forms the fibrotic scar, which, at least in part, serves as a barrier to regeneration. And so we've wondered whether we might be able to divert these scar-forming fibroblasts into the formation of new heart muscle cells. Now, over the past 15 years or so, our group and others have deciphered the core components of a gene regulatory network that drive the formation of the heart in organisms ranging from fruit flies to mammals. And at the center of this network of cardiogenic genes are 5 or so transcription factors schematized here. And we know from mutations and gain-of-function experiments of these transcription factors in many organisms that they're sufficient and in many cases, they're necessary and in many cases sufficient to drive the cardiac program. And so we've asked whether introducing any combinations of these or other factors into cardiac fibroblasts could promote their conversion into a cardiac fate. Now, to address that question, we first established a model system in which we generated transgenic mice that harbored in their genomes a reporter gene for green fluorescent protein linked to the DNA sequences for the alpha-myosin heavy chain gene, alpha-MHC, which is expressed only in the heart and not in any other cell types. We can isolate fibroblasts from the tail tip of these mice, put them in culture, and then infect them with viruses encoding various types of transcription factors, chromatin remodeling enzymes, or other molecules, and ask, can we find any that can activate the expression of the GFP reporter? And suffice it to say that from an extensive iterative screen of many transcription factors, drugs, microRNAs, etc., we ultimately settled upon a combination of 4 transcription factors. Shown here, you can just remember them as GHMT. These are conserved in organisms from fruit flies to humans and are dedicated to cardiac gene regulation in those organisms. And if these 4 factors are engineered into a virus and put into fibroblasts, they initially could switch on the GFP reporter, and that's how they were identified in this screen. Without getting into too much detail, this is what a typical assay would look like. If one isolates fibroblasts from the tail tip of these transgenic mice and separates them by flow cytometry, they express 0 alpha-MHC GFP, and they express 0 cardiac troponin T, a cardiac-specific structural protein. If we infect them with GHMT, you can now see that these cells have migrated to these positive channels and are expressed in quite robust levels of these cardiac markers. And this is what a typical fibroblast would look like after infection with GHMT. Here it's activated the GFP marker, and it's activated cardiac troponin T. This shows the quantification. There's no background in this assay, so tail-tip fibroblasts from mice do not express these markers at all. And when they're infected with GHMT, they upregulate these markers quite appreciably, but still there's much room for improvement. So we see roughly 20% of the cells become positive for these markers. This is what the cells look like. So these would be fibroblasts that have been transduced with the GHMT viruses in vitro. Initially, when stained, in this case for alpha-actinin, which is a cardiac structural gene, they show a diffuse distribution. They don't all have heart-shaped nuclei, but that was a good one. So they have diffuse cardiac protein expression in their cytoplasms, but with time and culture, their cytoplasms begin to organize, and ultimately, by about 4 weeks in culture, they show the classical striated sarcomeric appearance of adult cardiac myocytes, and a subset of these, a small subset, will beat spontaneously in culture. So based on these experiments and other work done by others, including Deepak Srivastava and many other groups, we were confident that we knew at least the primitive recipe of transcription factors that was sufficient to reprogram a fibroblast into a cardiac fate. And so we wanted to go ahead and do the ultimate experiment and ask, could we introduce these cardiogenic transcription factors into the heart in vivo and divert fibroblasts from the formation of a scar? Now, this is a complicated experiment, and it requires precise lineage markers so that one can trace the fate of a fibroblast in vivo. When we began these studies, the best-known fibroblast marker was a protein called fibroblast-specific protein, FSP1. That promoter driving a Cre recombinase, you can cross that into a mouse line that has a floxed Rosa lac-Z, so this Cre will activate lac-Z and turn fibroblasts blue. We then induce a myocardial infarction by tying off the left ascending coronary artery, left anterior descending coronary artery, and we inject with retroviruses encoding GHMT. Now, the viruses that we use for these experiments infect only proliferating cells, not postmitotic cells, so they'll infect the fibroblast population, but they won't infect cardiac myocytes. And the question is, can any of these labeled fibroblasts become blue cardiomyocytes? This is what a typical experiment looks like. This particular transgene combination is not as expressed above background levels in normal, unstimulated, stressed fibroblasts. So in a cross-section, you see little or no staining. If one induces a myocardial infarction and cuts the heart in cross-section, you can see all these fibroblasts forming the scar have activated this fibroblast-specific marker, so they're highly marked. There's no marking of cardiomyocytes with this marker. If you then inject into regions surrounding the infarct the virus encoding GHMT and wait roughly 3 weeks, what one sees is clusters of intensely blue labeled cells, which we believe arose from fibroblasts that were reprogrammed towards a cardiac fate by the GHMT viruses. At high magnification, this is what a typical heart would look like following this in vivo reprogramming assay. So here's two cardiomyocytes, A and B. A is obviously labeled with lac-Z from the fibroblast lac-Z marker, so we believe it came from the reprogramming process. B is unlabeled, so we believe that's a nascent, pre-existing cardiomyocyte. They're both stained in red with cardiac troponin T, a heart-specific marker, and they're connected by connexin 43, shown here, which is a gap junctional protein expressed specifically in the heart, suggesting that these two myocytes have connected properly. Now here's two small blue myocytes. We believe those both came from reprogrammed fibroblasts because they're labeled with lac-Z, and they're also connected by connexin 43. I won't go into the details, but suffice it to say you can dissociate these various cells and culture them in vitro. Here's an unlabeled cardiomyocyte, which is presumably a pre-existing one. It exhibits calcium transients and contractility with frequency characteristic of cardiomyocytes. Here are two myocytes that can be labeled with a cell-permeable dye for beta-galactosidase, so we believe they were reprogrammed, and they also show calcium transients and cardiac contractility. So we believe that these in vivo reprogrammed fibroblasts can adopt the properties of native cardiomyocytes. Now again, in these sorts of experiments, your ability to interpret them is only as good as your lineage markers, and so we wanted to generate an independent lineage marker for the fibroblast population. And so here we generate a second marker by inserting into the locus through homologous recombination a inducible Cre recombinase gene into this locus called TCF21, which the name of the gene is not particularly important. But we could label then these fibroblasts within the heart in vivo by crossing them to a tomato reporter. So all the fibroblasts turn red. So here you can see a typical histological section. The green striated myocytes are obvious, and nestled amongst them are these red cardiac fibroblasts. And so once again, we can activate tomato in these fibroblasts, induce a myocardial infarction in this heart, inject GHMT, and ask, can we find any tomato-positive cardiomyocytes? This is what an experiment looks like. So we've dissociated cardiomyocytes from these hearts following reprogramming and put them in culture. So here you can see two unlabeled cardiomyocytes in the background, and here's an intensely tomato-positive cardiac myocyte. We call these induced cardiac-like myocytes, ICLMs. You can do patch clamping on, this is a tomato-positive one, so presumably it came from a fibroblast, and here's a native endogenous myocyte, and they both have action potentials that look quite similar. So this is the last experiment I'm going to show you. That is to test whether in vivo reprogramming of fibroblasts using these cardiogenic transcription factors can enhance cardiac function. Now in these experiments, for those of you who aren't familiar with these approaches, cardiac function is measured by ejection fraction. This is the fraction of the volume of the blood in the heart that's expelled with each contraction. So in a normal mouse heart, unoperated, 70% of the blood would be expelled each time the heart contracts. And if you induce a myocardial infarction by tying off the coronary artery here, the heart loses contractility due to myocyte loss, and that drops to about 25%, and it stays at that level persistently. If we inject these hearts with virus encoding GHMT, what we've seen is that there's a dramatic improvement of cardiac function. They don't get back to normal, and I don't think we would expect them to do so, but they show remarkable improvement in cardiac contractility here at 6 weeks, 12 weeks, and we've now carried these animals out for over a year, and this is a permanent improvement of cardiac function following reprogramming in vivo with these cardiogenic viruses. Now, these experiments are clearly interesting. They're consistent with the in vitro data that we've generated on the reprogramming process, but they also raise, I think, some very important questions that we're currently wrestling with. The magnitude of functional improvement by in vivo reprogramming is much more dramatic than we would expect based on the number of cardiomyocytes that we can see to be reprogrammed from fibroblasts. And we're currently trying to understand what is the basis for this disparity. It could be that the reprogramming process is more efficient in vivo than we're able to visualize. We can only visualize those cells for which we can mark them with these lineage tracers, and maybe GHMT is reprogramming other cell types in the heart towards cardiomyocytes. But it's also possible that these cardiogenic viruses are acting through other mechanisms to enhance cardiac function. They may be themselves blocking fibrosis by inhibiting proliferation of fibroblasts. We know they're enhancing cardiac survival, and perhaps we know angiogenesis is robustly induced by these factors, and perhaps they're doing that more through some direct mechanism, and we're currently trying to sort that out. So lastly, what I've told you then today is that the heart is notoriously refractory to regeneration, and there is an enormous need to understand this process as a strategy for possible new approaches for heart repair. What I've told you is that we believe that there are at least 3 complex processes that are at play that dictate whether a neonatal heart can regenerate or an adult heart cannot regenerate, and those are the processes of fibrosis, inflammation, and regeneration based on cell proliferation. We think that we are beginning to get a foothold on some of these processes. I think we know some of the molecules, some of the genes, and some of the mechanisms involved, at least in idealized settings. And using that knowledge, we're currently identifying microRNAs, new drugs, and regenerative molecules that can augment the types of regenerative strategies that I've described to you today. And lastly, let me just close by acknowledging the people that have done this work. All the work I talked to you about on the role of macrophages in regeneration was carried out by a postdoc in our lab, Aaron Aurora. Gua Huang, who's a new faculty member at UCSF, has worked on the role of the epicardium in heart regeneration. And these are postdocs who have all taken new jobs at these institutions, and they contributed to all the other work that I described today. Thank you very much.
Thank you, Professor Olson. I think this will conclude the last session of today's meeting.